Hello everyone. Hello everyone, welcome back. So this is our revision class for the NPTEL course Fundamentals of Protein Chemistry, and this course is coordinated by Professor Dasgupta from the Department of Chemistry, IIT Kharagpur, and I am Beas Mukherjee, a research scholar from the Department of Bioscience and Biotechnology. And in this class we will be revising all the topics that we have already studied in our previous classes. So we have completed 11 weeks until now. So in today's class, we are just going to brush through some of these topics in the form of MCQ questions, and then we are going to deal with some of the MCQs that have come in the previous year's questions. Okay, for example, CSIR NET questions, and then GATE questions, DBT questions, all of these. Okay, so based on whatever we have understood from our classes, we are going to take up these questions and try to solve them. All right. So hopefully this class is going to be of some value to you. So let's begin with today's class. Okay. So first we started with amino acids, if you remember. So in the first two classes, we discussed about amino acids, and determining the PI of a dipeptide or tripeptide is one of the most important questions that you can expect in the NPTEL exam that's upcoming and in other competitive exams. Okay, so we will quickly brush through this once. So if you remember, this is a peptide, and how do you calculate the PI of the peptide? So for example, you have to take into consideration the pK of all the charged groups. So this N-terminal group is the amino group, and the C-terminal carboxylic group, both the the pKa of these groups you have to consider, and then you also have to consider the pKa of the COO minus group of aspartate and the COO minus group of glutamic acid. These are also two ionizable groups in this tripeptide. Okay, and then we have to check that at what pH, what is the final charge of this particular tripeptide. Okay, so for example, at pH 1, if you see, then the net charge is +1. How is that happening? Because at pH 1, if you take the charge of all of these different ionizable groups and if you add them up, you see that it is -3 and +1. So it becomes +1 charge when the PI is 1, pH is 1, so the net charge is +1 on this. Then if you see that when the pH becomes 2.0, the net charge is 0. And when the pH is 4, the net charge is -1, pH is 5, the net charge is -2, and when pH is 10, the net charge becomes -3. So now the PI is the two pKa values that are on either side of the pH at which the net charge is 0. So that means here you have to consider 2.19 and 3.86 for the PI calculation. So if you can see that the PI is calculated using these two values. Okay. Okay, so we'll move on. Now we'll try to solve some very simple questions. These might already have been there in your previous slides, so you can actually try to solve this. Okay, so you can type in the chat box whatever you think is the right answer. So the question is, which technique is suitable for investigating the kinetics of protein folding and unfolding transitions? So option number A is circular dichroism spectroscopy. Option number B is isothermal titration calorimetry. Option C is differential scanning calorimetry. Option D is FRET, that is fluorescence resonance energy transfer, and option E is X-ray crystallography. So which of these techniques do you think would be most appropriate to study the protein folding and unfolding transitions? Okay. For the next, okay, someone is joining. Okay. For the next question, it is, What method is suitable for determining the stoichiometry of protein complexes? Most suitable for determining the stoichiometry of protein complexes. So A is analytical ultracentrifuge, B is nuclear magnetic resonance spectroscopy, C is size exclusion chromatography, D is ITC, and E is FRET. So you can answer in the chat box and then we can look at the right answers. Okay, so let's look at the right answers. So okay, I think there is a mistake in the second question. Okay, so for the first question, the right answer is differential scanning calorimetry. Okay, but for the second question, your analytical ultracentrifuge is a good answer, and size exclusion chromatography is also a good answer. So what would be most appropriate for determining the stoichiometry of the protein complexes? So size exclusion chromatography and analytical ultracentrifuge would be more appropriate answers compared to ITC. Okay. Now we move on to the next question. So the next question is a match the following question. There are different chromatographic techniques and different column material, and you have to match these. Okay. So the first one is affinity chromatography, then gel filtration chromatography, cation exchange chromatography, hydrophobic interaction chromatography. And the column materials are silica gel, CM cellulose matrix, NINTA matrix, and biogel P300, and then DEAE separose matrix. So which do you think match? So we'll look at the right answer. So the right answer is option number C, which is 1 is 3. So affinity chromatography, of course, you need NINTA matrix. And then for gel filtration chromatography, it is biogel P300. For cation exchange chromatography, it is carboxymethyl. No, 1 second. Yeah, it is carboxymethyl cellulose. And for hydrophobic interaction chromatography, it is silica gel. Okay. Now, the next question is, which, in terms of metabolic pathways, which amino acids are considered glucogenic? So, select the ones that apply. So, what is glucogenic and what is ketogenic? So, we will discuss that very soon. Okay. So, some of the amino acids are glucogenic and some of the amino acids are not glucogenic. So, which one do you think is a glucogenic amino acid? Okay, so all the three of these are glucogenic amino acids. Okay, A, B, and C, all the three of them are glucogenic amino acids. Now, the difference between glucogenic and ketogenic amino acids is given for your reference. So, what is this? The amino acids are degraded to acetyl coenzyme A, and acetoacetyl coenzyme A are used in the citric acid cycle, but mammals cannot synthesize glucose from acetoacetyl coenzyme A. The fact is the source of distinction between glucogenic and ketogenic amino acids. Okay, so glucogenic amino acids can be converted into glucose with oxaloacetate as the intermediate, but ketogenic amino acids cannot be converted into glucose. So some of the examples of the glucogenic and ketogenic amino acids are: glucogenic amino acids are alanine, aspartate, glutamine, serine, and arginine. And some of the ketogenic amino acids are leucine, lysine, tryptophan, phenylalanine, and isoleucine. Okay. So we will look at this in a little more details. So if you look at this metabolic pathway, you see that how these different amino acids are getting converted into different products of the Krebs cycle, and these are actually being used for uh, generation of glucose and generation of ketone bodies, and that is how they become glucogenic and ketogenic amino acids. Okay. Now, there is a question for your practice. The folding and stability of a protein structure are influenced by various interactions and modifications. So consider a hypothetical protein, protein X, which has 100 amino acids. Which of the following interactions are so the following information is available about it. Okay, so this hypothetical protein X has 200 amino acids, and the following information is available about it. So protein X has two domains. Domain A is residue 1 to 100, and domain B is residue 101 to 200. So domain A forms an alpha helix rich structure stabilized by intrahelix hydrogen bonds and hydrophobic coat packing. Domain B consists of a beta sheet structure stabilized by hydrogen bonds between adjacent strands and disulfide bridges between the cysteine residues. Protein X undergoes post-translational modification involving phosphorylation of serine residues in domain A, acetylation of lysine residues in domain B, and glycosylation of asparagine residues in both the domains. So the protein is found to denature completely when the pH drops below 5 but retains partial structure at pH 6 due to persistence of ionic interactions involved in the charge residues in both the domains. Okay, so that is the information that is given to you about protein X. Now let's see what is the question. So now the question is, which of the following statements is most accurate regarding protein X? Okay, now the answer is unfortunately already there on this slide. So no problem, we'll look at it. So now option number A is phosphorylation of serine residues in domain A enhances alpha helical stability by increasing the intrahelix hydrogen bonding. Acetylation of residues in the domain B is likely to weaken the ionic interactions within the beta sheet structure by reducing positive charge. Disulfide bridges in domain B are most likely to break at pH 6 due to increased competition from the protonated cysteine residues. Glycosylation of asparagine residues in domain A increases the flexibility of the alpha helix by disrupting the hydrophobic interactions. So out of all of these different options, option B is the most appropriate. Okay, this is the most correct option. Now the explanation is given to you. Why is it the most appropriate answer? So option A is incorrect because phosphorylation typically introduces a negative charge to serine residues which can disrupt rather than enhance the intra-helix hydrogen bonding. Okay. Option B is correct because acetylation neutralizes the positive charge of the lysine residues which would weaken any ionic interaction. For example, the negatively charged side chain or phosphate group. Okay. So option C is incorrect because disulfide bridges are covalent bonds and are not typically affected by changes in pH until extreme conditions are reached. Option D is incorrect because glycosylation often increases the structural stability rather than flexibility, especially in the structured regions like an alpha helix. Now, disulfide bond formation is dependent on thiolate concentration, which makes it pH dependent. The optimal range for the disulfide bond formation is between 3 and 8. So pH 3 and 8. So disulfide bonds can break at the critical pH of 10.5. So that is a very interesting point about disulfide bonds which you must remember. Okay. Now this explains why the option B was a correct option. Okay. Now we will move on to the next question. So now these are previous year's questions. Okay, so all of these are previous year's questions. They have been taken from different exams, exam question paper, and that is mentioned. Okay, so for example, this question is CSIR NET 2023 question. So this is how you will be able to go back when you're solving these questions from these papers. You'll be able to actually relate. Okay. So we'll start with the questions. So polypeptide was subjected to the following treatments with the indicated results. So if you remember, we had talked about all of these different ways in which the peptides are broken up. They are chopped off, right, by different kinds of agents. So see what is happening here. Acid hydrolysis is happening. Acid hydrolysis is going to tell you about the composition of the peptide, okay, or the polypeptide. So whatever amino acid is there, that you get to know because everything is going to get chopped up, but you will not know the sequence. Now aminopeptidase M. So what is that? It is not giving any fragments. Now next is carboxypeptidase A plus B. So if you remember in our slides, we had talked about carboxypeptidase A, B and C. Right, so both A and B are given together. So now no fragments. What does it mean by no fragments? So that means that there is some amino acid at the end terminal of the last amino acid which is not cleavable by this carboxypeptidase A and B. So that is why it is not giving any fragment. Okay. Now the trypsin followed by Edmond degradation of separated products. Now Edmond degradation and trypsin digestion, these are also done. So that is giving you this fragment, and then you get this other fragment also. So which one of the following represents the primary structure of the peptide? So you can understand that based on your understanding of all of these different reactions, you will have to predict what was the initial structure of the peptide. So this is how questions come, and this is how you're supposed to solve them. Okay. So all the three different options are linear and one option is for a cyclic peptide. So let us look at the answer. So see, the answer is 4, the cyclic peptide. And now the answer, the explanation to this is also been taken from this particular paper. Okay, so that you can understand how to approach questions, how to understand how to solve the questions. Okay. So carboxypeptidases and aminopeptidases are exopeptidases that remove terminal amino acid residues from the C and N termini of the polypeptides respectively. So carboxypeptidase A cleaves the C terminal peptide bond of all amino acid residues except proline, lysine and arginine. Alright. Now carboxypeptidase B is effective only when arginine or lysine are in the C terminal residues. Carboxypeptidase C acts on any C terminal residues. Now aminopeptidases catalyze the cleavage of amino acid from the amino terminus, so N terminus of the protein. Aminopeptidase M catalyzes the cleavage of all free N terminal residues. Okay. So since this is not giving you any cleavage, how is that possible? That means there is no free N terminus, correct? So that is why you're not getting cleavage. Now, that is why this product is actually a cyclic peptide. Okay, it's a cyclic peptide, and that is why this N terminal carboxypeptidase was not giving you any product. So now we have a question from CSIR NET 2023 once again. So which of the following statements is correct in the context of erythromycin mediated inhibition of protein synthesis in bacteria? So erythromycin inhibits protein synthesis at the step of elongation, but it nonetheless allows translation of the first few codons. Erythromycin inhibits protein synthesis at the step of elongation, and it prevents its formation of even the first peptide bond. Erythromycin inhibits the formation of translation initiation complex. Erythromycin is toxin to bacteria because it results in the initiation of protein synthesis with elongated tRNA. Now from here you can understand that the questions that they are asking are in depth, right? So now to understand exactly how much you should read for a particular topic, what is very important is that you solve these questions, as many as you can, because only then you will have a better idea about approaching questions in an exam. Okay, so what is the solution to this question? Let's see. The solution to this question is option number 1. Okay, so chloramphenicol binds to the 50S ribosomal subunit and blocks the peptide bond formation through inhibition of peptidyl transferases, but does not affect the cytosolic protein synthesis in eukaryotes. Tetracycline binds to the 30S ribosomal subunit, interferes with amino acid tRNA binding. Erythromycin binds to the 50S ribosomal subunit and inhibits protein chain elongation. Okay, that is important. Next, fusic acid binds to the EFG and blocks translocation. Cyclohexamide blocks the peptidyl transferase of the 80S ribosome but not that of the 70S bacterial and mitochondrial chloroplast ribosomes. Okay, so this is how you need to be very specific about what you're studying, how you're studying it, and how you're answering the questions. Okay, so we have the next question here. This is also from CSIR NET 2023. So the following statements are made about digestion of proteins and enzymes involved. Chymotrypsin does not generate peptides with the C-terminal neutral amino acids. Trypsin generates peptides with C-terminal basic amino acids. Carboxypeptidase B acts on aromatic amino acids. Carboxypeptidase A employs zinc ion for hydrolysis. The brush-bordered enterokinase has no polysaccharides attached to it. The final digestion of amino acid occurs in the intestinal lumen, the brush border, and the cytoplasm of the mucosal cell. So which one of the following options represent the combinations of all correct statements? So this question is incorporating a few different topics. Besides this peptide digestion, there are also other topics, and this is how the questions are expected in your exams. Okay, there will be questions combining the knowledge of 4 to 5 different topics, and that is why you will choose to attempt the questions only about which you are 100% sure. Okay, so let's say you haven't studied much about these few topics, so then you skip the question because there will be a lot of different questions and you will have to only choose and attempt only the ones about which you're confident. Okay, so for this question, let's see what is the right answer. So right answer for this question is option number 4. So that says B, D and F are correct. Okay, so for trypsin, chymotrypsin, pepsin, elastase and thermolysin, the site of the cleavage is given for you for your knowledge, and carboxypeptidases and aminopeptidases are exopeptidases that remove terminal amino acid residues from the C-terminal and N-terminal polypeptide respectively. Carboxypeptidase A cleaves the C-terminal peptide bond of all amino acid except proline, lysine and arginine. Carboxypeptidase B is effective only when arginine and lysine are in the C-terminal residues. C acts on any C-terminal residues, right? So, and whatever we studied in the previous one is again given over here. So, this is how you will be able to understand that D, D and F are the right options. So, we'll move on to the next question. You can try to solve this. I'll be back within a minute. Right, so we'll carry on. Okay. Now, the question that they're asking is some properties of enzymes are listed in column X and their kinetic expressions are listed in column Y. So, the enzyme properties are given. For example, specific activity, turnover number, Michaelis constant, catalytic efficiency. Okay, and expressions are given as kcat by km, substrate concentration at which velocity of the enzyme is equal to half of Vmax, Vmax by moles of enzyme, Vmax by protein concentration. So now from here you will have to say that which expression corresponds to which enzyme property. So when we were talking about these different enzymes and their activity and their different characteristics, I had told you a million times that these are very important questions for your exam. So please remember what is the meaning of what property of the enzyme. So which expression goes for which property. Okay, so now they're asking you to match this. Okay, this is simple if you understand this. So now let's try to solve the question. So we look at the right answer. Okay, and the right answer is option number 3. Answer number 3. Okay, which tells you that the specific activity of the enzyme is defined as the amount of enzyme activity measured in enzyme units per unit of protein. So it is a measure of the enzyme's catalytic efficiency and is often used to assess the purity of the enzyme preparation. So specific activity is calculated by dividing the total enzyme activity by the amount of enzyme present in the sample. It provides a measure of the concentration of the active enzyme molecules in relation to the total protein content. Okay. Now we come to the next question. So given below are the terms related to various experimental technique. Now from here you can understand that in the same question paper, there are so many questions already there that we have covered in this course. So you will be able to solve all of these, okay, if you understand the course properly. So given below are the terms related to various experimental techniques. So the technique is given and the application is given. Okay, mass spectrometry, pulse field gel electrophoresis, isoelectric focusing, and tactile. So the applications are given: separation of whole chromosome, separation of isozyme, single molecule real-time sequencing, and identification of post-translational modification of proteins. So which one goes for which? Okay, so we'll look at the right answer. Option number 1A is the right answer. So A is for 4. Mass spectrometry is definitely useful for determining post-translational modifications of proteins. Okay. B, pulse field gel electrophoresis. This is useful for studying whole chromosomes. Correct. Isoelectric focusing is very useful for separation of isozymes. And PacBio is of course used for single molecular real-time sequencing. Okay, or smart sequencing. Now we have a few more questions. Let's try to solve them. The hexapeptide P is an isoelectric, has an isoelectric point of 6.9. So now we have come to the GATE BT 2019 questions, right? So let's try to solve them. So hexapeptide P has an isoelectric point Pi of 6.9. Hexapeptide Q is a variant of P that contains valine instead of glutamate at position 3. So the two peptides are analyzed by polyacrylamide gel electrophoresis at pH 8. Which one of the following statements is correct? So P will migrate faster than Q towards the anode. P will migrate faster than Q towards the cathode. Both P and Q will migrate together. Q will migrate faster than P towards the anode. Right. So for the second question, it is group 1 list spectroscopic methods and group 2 list the biomolecular applications of these methods. So match the methods in group 1 with the applications in group 2. So infrared spectroscopy, circular dichroism spectroscopy and NMR spectroscopy. So identification of functional groups, determination of the secondary structure, estimation of molecular weight, determination of the 3D structure. This is simple, very straightforward, very direct. Let's see what happens. What is the right answer? Okay, so the right answers are for the first question, the right answer is A. So what is happening that the hexapeptide Q is a variant of P that contains valine instead of glutamic acid, and valine is a what? Hydrophobic amino acid. It is not charged. Glutamic acid was having extra negative charge, right? So when the two peptides are analyzed by page at page 8, so what is happening at page 8? Your P will move faster than Q towards the anode. Why is that so? Because COOH group will have left this COOH and it will leave the H plus ion, it will become COO minus, right? So it will have that extra negative charge, because of which it will move faster towards the anode. Why anode? Because anode is positively charged and this is negatively charged, right? So it's an anion and it is moving towards the anode. And for this question, the second question, the right answer is C. Why is it C? Because your circular dichroism is always going to give you information about secondary structure. NMR is going to give you information about 3D structure, and infrared can be used for the identification of the functional groups. Okay. Now, so how many questions do we have? We are at which one? 27 now, and we have a few more left. Okay, great. Right, so this is again GATE BT 2019. Okay, so antibody producing hybridoma cells are generated by a fusion of. So we were talking about this in the last class, right? So T cell with myeloma cell, B cell with myeloma cell, T cell with B cell, macrophage, and a myeloma cell. You definitely know the right answer to this, right? So we were discussing about this in the previous class. Now, which of the following factors have the fidelity of DNA polymerase in the polymerase chain reaction, which affects the fidelity? What is fidelity? Fidelity means that, let's say there is an A at this particular position, an adenine. So it is going to add a thymine in the complementary strand that it is making. So that is fidelity. It will not make mistakes. Okay. And it is also not going to hopefully follow. So that is processivity. So which of the following factors affect the fidelity of the DNA polymerase in polymerase chain reaction? Mg2 plus concentration, pH and handling temperature. Okay. So let's see what is the right answer for this. So the right answers are option number B, which is B cell and myeloma cell, and option number D, which is P, Q and R. So all of these are important factors for affecting the fidelity of the DNA. Okay. Now GATE Excel 2020, 2019 and 2023. So this question was actually repeated in 2023 as well. So which of the following techniques cannot be used to determine the sequence of a novel protein? So de novo sequencing by ESI-MS. So tandem mass spec. Okay. Sanger sequencing, Edmond degradation, peptide mass, fingerprinting. Very direct question. For a novel protein, which of these techniques cannot be used for determining its sequence? A protein has 7 cysteine residues. The maximum number of disulfide bonds of different combinations that can possibly be formed from these 7 cysteine residues is. This is a NAT, numerical aptitude test. So the right answer is given here. Okay. Sanger sequencing cannot be used to determine the sequence of a novel protein because Sanger sequencing is for DNA, right? It's for nucleic acids. Now, for question number 33, the right answer is 48. Okay. How is 48 coming? I think we have discussed this in our previous classes. So you can refer to that. Okay. So now, here they have in fact explained how this is coming. So when there are 4 cysteine residues in a protein, they can form 2 intramolecular disulfide bonds in different ways. The number of different ways can be calculated in this way. So see, first is determining the number of possible disulfide bonds. So the formula is given to you in and then you can actually do it this way. So n factorial divided by r factorial into n minus r factorial, where n is the number of cysteine residues and r is the number of disulfide bonds. So in this case, the number of cysteine residues is 4 and the number of disulfide bonds is 2. So the number of possible disulfide bonds is 6. Okay, now determine the number of different ways in which this disulfide bonds can be formed. So it can be between 1 and 3, it can be between 2 and 4, 1 and 2, 3 and 4, 1 and 4, 2 and 3, right? So that is 3 different ways. The number of different ways that the disulfide bond can be formed is 3. So in conclusion, when the protein contains 4 cysteine residues, the number of different ways they can form simultaneously 2 disulfide intermolecular bonds, intramolecular bonds is 3. Okay. Now, what happens if that is if there are 8 cysteine residues can form disulfide bonds, so then there are 4 different ways in which they can form the disulfide bonds, right? So 4 different bonds can be there at the same time, and how many different ways? Lot more. So understanding the disulfide bonds and then choosing the pairs and calculating the pairing, so you can read all of these, and from here you can get to know how many disulfide bonds are getting formed and how they are getting formed. What are the different possible combinations? So here also it is explained that formula for the combination. Okay, total pairing is explained to you. Calculating each combination, putting all of that together, it is saying that if 8 cysteine residues are there, it can form disulfide bonds in 105 different ways. Okay, now we will come to the next one. Which of the following techniques can be used to separate proteins according to their molecular weight from a mixture of proteins? Okay, according to the molecular weight. So, of course, you know there are different methods that you're familiar with. Okay, the right answer is B, C, and D. Right. So, of course, size exclusion chromatography, SDS page, and sucrose density gradient centrifugation, all of these are useful methods for separation of proteins based on their molecular weight. Okay. So that's all for today's class. This is how you can go through all the different questions from different exams, and you can expect these questions in the NPTEL questions question paper as well. And all the best for your exams. Okay, all the best for your preparation. So all the different links from which I had taken all of these different questions are explained. The explanation of the different answers are also given here. Okay, the sources are all cited. So thank you for your attention. Thank you for participating in the classes for all these weeks. Okay, have a very good day. Thank you. So if you don't have any more questions, I'll be ending the class now. Okay, thank you so much for your attention. So do you have any more questions? Okay, thank you so much. So we'll end the class now.